fathom what changes this small virus would be bringing upon in our day-to-day lives. The last 2 years, it has brought on a large scope of changes, including fast tracking of diagnostic tests, rollout of large-scale technological applications, an abundant research activities, wearing PPE as a normal part of our clothing, working from home, attending online seminars and workshops. All this has become the new norm. So keeping this growing importance and harsh reality of COVID-19, Pushpari Research Center, along with MacFirst, is catering this edition of Bioradiance as an extravagant feast for you all, gathering under the same roof brilliant minds, great academicians, and credible researchers to keep us abreast with the latest in COVID vaccines and public health impact of COVID. Hello, I'm Dr. Shirley Anthony, and I'll be your host for the evening. Do feel free to post your queries in the chat box and also in the Facebook live stream. Kindly mute yourselves and follow the etiquettes as the new norm of online webinars. So first, I'd like to share my screen. So going on to introduce our first speaker for the day is none other than Gagandeep Madam. Madam is the Professor of Microbiology, Division of Gastrointestinal Sciences in Christian Medical College, Vellore. Madam did her MBBS, MD and PhD from Christian Medical College. You can see her abundant qualification. She has done MRC PPAT.
Dr. Shirley?
Yes.
Dr. Shirley, your screen is not right, I think. It's just showing the table.
Okay. Just a second. Yeah, can you see now?
Yes, you can go into presentation mode. Yes.
Yes.
Okay.
Sorry for the glitch. Okay, so as you can see, Madam has had abundant educational qualifications, including she's done her MRC PATH, Efficacy PATH from Royal College of Pathologists. She's got fellowships from various national and international agencies, and to cap it all, she's the Fellow of the Royal Society of London. Madam has been awarded a lot of awards in the national level, international level, and currently Madam has got research fund more than $500,000 US, and Ma'am's lab is the WHO reference laboratory for rotaviral surveillance, and she's got funding from Fogati International Center, Bill and Melinda Gates Foundation, Department of Biotechnology, Wellcome Trust. So Ma'am is the ideal candidate to tell us about COVID vaccines and variants, and her publications are more than 330, including there are extra review articles, case reports, and book chapters. And Ma'am, in fact, has even written a book recently. So on to you, Ma'am.
Thank you very much for the introduction, Shirley, and thanks to everyone for inviting me to participate at this meeting. What I'm hoping to do over the next half an hour or so is to talk to you about COVID-19 vaccines, vaccination, and variants. And it's impossible to cover everything in just one talk. So what I'm going to try and do is to share with you key areas, and then I hope we will be able to spend some time on discussion of questions that any of you might have. I know that I certainly have a lot of unanswered questions in the areas of vaccines and what will happen next with the variants. So I think when we think about infectious diseases and vaccination, one of the things that's really important to understand is that in many ways for vaccination, what we are trying to do is mimic what happens in infection, but to do it in a way where we produce a protective immune response without making the person sick. So in order to do that, usually the traditional approach to vaccines has been to take a whole organism or a part of an organism and then find a way of weakening it or killing it and then give it so that you don't get disease, but you get the immune response. Sometimes we don't use the organism directly, we use a related organism. And an example of that is the very first vaccine that we have, which is where we use the cowpox virus to protect from smallpox. We also use BCG to protect us from tuberculosis. So related, not identical viruses and bacteria can also give us protection. Now, when we think about infections that are mucosal infections that come in through the gut or the respiratory tract, there are two kinds of immunity, adaptive immunity, that become important. One is systemic immunity, which is where you have antibodies and white blood cells that circulate and can reach practically every site in the body. But you also have mucosal immunity on the surfaces, and this is mostly mediated with the presence of IgA. Now, when we get infected with an organism that comes in through the respiratory tract and the gut and the genital tract, we usually make a systemic immune response as well as a local mucosal immune response. Now, if we give a vaccine that is given by the same route by which infection enters, we would wind up with systemic or mucosal immunity. But we know that with the vaccines that we have, most of them are being given through intramuscular injection, which means that when we get vaccine-induced immunity, most of the immunity that we are getting is systemic immunity. And that's one of the reasons why people are working on nasal vaccines, because nasal vaccines have the potential to give us good mucosal immunity also. The other thing to remember is that even though we use a lot of vaccines for SARS-CoV-2, all vaccines are not identical. They come on different platforms, and we'll discuss those platforms. But in one thing that will have been clear to everybody is that many of the vaccines that we work with currently don't focus, except for the inactivated vaccines such as Covaxin. Most of them don't focus on the whole organism. Most of them are focused on a single protein or only part of that single protein, and that protein is the spike protein of SARS coronavirus 2. Now, the spike protein is a trimeric protein, 3 components that wrap around each other to produce the spike. And the spike, each part of the spike has 3 components as well. It has the S2 domain, which is the globular domain at the top. S1 domain, which is the globular domain at the top. The S2 domain, which acts as the stalk. And on the top of the S1 domain, you have the receptor binding domain, which is the area that interacts with the ACE2 receptor. Now, one thing that is important to understand is that proteins tend to be stiff or floppy, but they can change shape. And when they need to interact with another protein, then the shape that a protein may have may be different from the time when it does not have that interaction. So with the receptor binding domain of SARS-CoV-2, there are 2 positions in which it is found. One is called the lying down position, and the other is the standing up position. In the lying down position, it doesn't bind well to the receptor, and this lack of binding or the position being changed can lead to immune evasion. And in the standing up position, you get really good binding to the receptor, and you get a high level of immune recognition also. So with the spike protein, what most people are doing when they're making vaccines to the spike protein is trying to use a consensus sequence of the spike. And in some of the vaccines, there has been mutations introduced into the receptor binding domain of the spike protein in order to stabilize the spike so that it forms a structure to which antibodies can be induced that bind really well. So we are trying to do somewhat better than nature in some of the vaccine designs that we have. So what are all the platforms that we have for vaccines? We have the platforms that are the technologies that we are familiar with, where we use the whole virus, and these vaccines can be live attenuated vaccines or inactivated vaccines. And in India, Serum Institute of India and Indian Immunologicals are working on live attenuated vaccines. These are vaccines where mutations are deliberately introduced into the genome in order to make sure that the virus does not replicate very well and does not cause disease. These are also called codon D optimized vaccines. And then we have inactivated vaccines. In India, we have the example of Covaxin, but China also has Sinovac and Sinopharm vaccines, which are also inactivated vaccines. And with an inactivated vaccines, in general, you usually give an adjuvant because you want to try and maximize the immune response as much as possible. In the case of Covaxin, we use an adjuvant that it has been developed quite recently, and this is the first human vaccine itself. In addition to using whole viruses, as I mentioned, we can use parts of viruses, and these can be in the form of virus-like particles, or they can be as separate proteins. An example of a vaccine where you have virus-like particles is the Novavax vaccine, where the spike trimers actually join together to form a shape that looks a bit like a flower. And this essentially is not exactly a virus-like particle that looks like the native virus, but it does look like a virus. In addition to that, we have recombinant DNA vaccines, and an example of a vaccine that is recombinant DNA and being made in India is the vaccine being made by Biological E, called Corbevax, for which the Indian government has already placed an order. Most of these vaccines are given with some adjuvant or the other. Then we come to the new generation of vaccines where you don't give the virus and you don't give a part of the virus, but you just send in the message for how to make a viral protein. And these are vaccines that are vectored or nucleic acid vaccines, either DNA or RNA. Among the vectored vaccines in India, currently we have the AstraZeneca or Covishield vaccine, where an adenovirus vector is used. In the case of Covishield, we use a chimpanzee adenovirus. And the reason for using the chimpanzee adenovirus is because humans won't have antibodies to the chimpanzee adenovirus. There is also the Johnson & Johnson vaccine, which is also going to be made by Biological E, and that uses the Ad26 vector adenovirus, a human adenovirus, but not one that is commonly found in humans. Then we have the Sputnik vaccine, which uses a combination of two different adenovirus vectors, Ad26 and adenovirus 5. And these two, again, adenovirus 5 is reasonably common in humans, but Ad26 is rare. And the idea of using the combination here is that Ad26 antibodies will be produced, and then if you give Ad5, you will get a boosting of the response because the antibodies will not interfere with adenovirus 5 replication. There are also, beyond adenoviruses, there are also other vectored vaccines that are being produced. And some of those vaccines have succeeded or are on the way to success, while others, like the vesicular stomatitis virus vaccine that was based on the Ebola vaccine platform that was being developed by Merck, has actually failed. If we look at DNA vaccines, Zydus Cadila has the first human DNA vaccine that has been licensed for an infectious disease anywhere in the world. Prior to this, DNA vaccines were available, were registered, that these DNA vaccines were used in animals and in fish. So to treat infections of fish, or to treat cancer in dogs, or in horses and pigs to try and prevent infectious disease or promote growth. So DNA vaccines are a technology that has been around for 30 years and in use, but not in humans. For vectored vaccines, actually the first vectored vaccines that we had were for Ebola, and those were fully licensed only about 2 years ago. Then we have RNA vaccines, which have been a standout success of the pandemic. Before this, there were no RNA vaccines that were in use in humans, animals, or anywhere for anything, though a lot are being experimented with. And while DNA vaccines have been around for 30 years, RNA vaccines have only started to be worked on in the last 15 years, because RNA itself is a very unstable molecule, breaks down very quickly in the human body. So therefore, we had to find a way of protecting RNA. And that was done by formulating lipid nanoparticles that could protect RNA before it got into the host cell. In addition to that, RNA is quite an inflammatory molecule, and it had to be figured out how to reduce the inflammation that is produced by RNA before vaccines could be worked on. Now we have two successful mRNA vaccines, the Pfizer and the Moderna vaccines, that are in use in many parts of the world. And in India, we have the Genova vaccine, which is currently in human testing. So where are we globally at the moment? We have about 100 vaccine candidates that are in preclinical studies, about 55 that are in the first phase of human testing, which is where we take small numbers of people, 10 to 100 people, give them the vaccine and try and figure out, is the vaccine safe and have we got the right dose. Then we have about 45 vaccines which have moved beyond phase 1 into an expanded safety and immunogenicity trial, a phase 2 trial. And in these trials, which usually have somewhere between 100 and 1,000 participants, we look again at safety, but also look to see whether the vaccine is inducing a robust immune response, mostly measured as antibodies. Then we have phase 3 clinical efficacy trials, and there are 34 vaccines in phase 3 trials. And these are trials where we try to figure out, if I give the vaccine to half the people and don't give the vaccine to the other half, how much reduction in disease will I see? So phase 3 trials are going to be increasingly difficult to do, because whenever we think about clinical research, we have to make sure that we are following standard of care to look after people. So if you are in a country where vaccines are licensed and being used, then it becomes actually very difficult to do a study where you give only half the people vaccine and don't give it to the other half. So you have to think about ways of testing vaccines that go beyond placebo-controlled trials. You might have to give people another active vaccine that is licensed, and then do a comparison to show that there is no difference between the two groups. But these studies then become very big and complicated to do. Anyway, through all of the preclinical and the clinical phase testing that has happened, we have currently reached a stage where we have 13 licensed vaccines that are in limited use. And then we have full use with emergency use authorization of 8 vaccines. And recently, the Pfizer vaccine has been given a full approval. Now, when we hear all the talk about vaccines, we hear about breakthrough cases, we hear people talking about variants, talking about the need for booster doses. I think it's important for us to remember where we were last year. In May of last year, WHO published a target product profile, and we were thinking at that time that SARS-CoV-2 vaccines would work rather like vaccines for influenza, which meant that if you had a vaccine that had even 50% efficacy, that would be considered a pretty good vaccine. So what WHO was looking for was a vaccine that needed 3 doses, that would be usable in adults, that would give protection for 6 months, that would have 50% efficacy. And as we know now, practically every vaccine that we have has met and exceeded that target. So which are the vaccines with efficacy data? For mRNA vaccines, we have Pfizer and Moderna. For the vectored vaccines, we have the AstraZeneca, the Gamaleya, and a Chinese vaccine, CanSino Bio, which also uses the Ad5 vector. Sinopharm, Sinovac, Covaxin are inactivated vaccines. There are 2 vaccines that have come out of Cuba, and Cuba is in fact taking its age of vaccination down to 2 years of age. And the vaccines that they have are a subunit vaccine and a conjugate receptor binding domain vaccine, where the RBD is conjugated to tetanus toxoid. We also have phase 3 efficacy data on the Novavax vaccine, but this is not a vaccine in use anywhere because they have not applied for approvals in the US. They're having some issues with some of their manufacturing data. This is a vaccine that is being made by Serum Institute of India as Covovax, and clinical trials are ongoing in India, and it's even possible that we might have approval for Novavax's vaccine in India before there is approval in the US. And then recently, based on interim data, we had an approval given for the DNA vaccine made by Zydus Cadila. Excuse me. So now if we look at what results in protection, this looks like a complicated slide, and immunology slides always are. These are examples of the mRNA vaccine on top, the adenovirus vectored vaccine below. Both of these vaccines act by delivering a message inside the dendritic cell or an antigen presenting cell. What that cell then does is it takes the message, it reads the message, it makes the spike protein, and then it displays the spike protein on the surface or releases it outside the cell. When that happens, an immune response that is mediated by cytokines, chemokines, and interferons is initiated. This then allows for the antigen presenting cell to present the antigen, which is part of the spike protein, in conjunction with an MHC class II molecule to the T cell. The T cell receptor recognizes this signal, and this results in stimulation of the T cell. T cells can then stimulate T follicular helper cells, which then go on to support B cells in their ability to make antibodies against the spike protein. Some of these B cells will make antibodies as plasma cells. They'll become little antibody factories. Some of them will go on to become memory B cells. In the T cells, the cell populations that are set up, one is the follicular helper cells that will be responsible for the T and B cell interaction. Then you have the effector T cells, which can act by directly recognizing infected cells or by secreting more cytokines and chemokines. And some of the T cells will go on to become memory cells. Now, one of the things that comes up very frequently when we talk about vaccines, and particularly when we talk about what's happening in India today, is that a lot of people are going out and measuring their antibody responses after they have been vaccinated. And what they find is that some people have very low levels of antibodies. Other people have antibodies that start out high, that come down after a while. And then there are always lots of questions about whether they need to take a booster dose to maintain their antibody levels at a high level. And here I'd just like to point out that the point in having memory cells is that if you have memory cells, when you get infected, you can very quickly make antibodies again. So as long as you have generated a good immune response to your first infection or vaccination, you're not likely to need to maintain your antibodies at a high level. Now, we know that antibodies protect. We have laboratory studies that show us that antibodies neutralize SARS-CoV-2. We have studies from animal models, and shown here in these graphs is data from non-human primates, where it was shown that if you take non-human primates and you give them antibodies from an infected or vaccinated animal, so you transfer serum from an infected or vaccinated animal that has antibodies, what it will result in is a suppression of replication in protection of the transferee. The primate that has received the serum will not allow the virus to replicate, and this happens in a dose-dependent manner. So you get protection just through the passive transfer of antibodies. We also know from human data that has come recently that monoclonal antibodies work both for prevention and for treatment of severe SARS-CoV-2 infection in people who have not had prior exposure. So we know that antibodies work, and we know that if we generate a memory response, then when we encounter the virus after vaccination, we will be able to mount an immune response quickly. Now, the other question is how much antibody is enough antibody? I think one of the things that's really important to recognize is all these commercial tests that we look at that people are running off to get are really tests that measure binding antibodies, IgG that binds to the spike protein. And while IgG that binds to the spike protein correlates with the neutralizing antibody response, it is not a perfect substitute for the neutralizing antibody response. And we know from other viruses that it really is the neutralizing antibody response that matters. And shown here in these graphs is data that has come from multiple different studies that have looked at how much neutralizing antibody was made in vaccine trials. And what they've done here is they've taken the blue dot here with the bars. They've taken convalescent sera because every vaccine trial, because there wasn't an international standard available, took serum from people who had recovered from infection and used that in a control in their studies for neutralizing antibodies. So what has been done here is to look at neutralizing antibodies generated by different vaccines measured in comparison to convalescent sera. And then that is compared to protective efficacy shown by that vaccine in phase 3 studies. So what we can see here is that the mRNA 1273, the Moderna vaccine, gives very good protection and has high levels of neutralizing antibodies, about 4 times what you find in convalescent sera. And if you look at the other end of the graph, you see much lower protection with CoronaVac, which is an inactivated vaccine, but you also see a much lower level of neutralizing antibody response. So in general, what these graphs show is that neutralizing antibodies correlate with protection, but you don't know where there is a perfect cutoff. So you can't say, if you have neutralizing antibodies that are double what you will see in convalescent sera, then you are guaranteed protection, because we really don't have an understanding of what that is. And this is true for many mucosal infections. We frequently wind up in a similar sort of situation where you don't have a clear understanding of what level of antibodies might be protective. So in India today, we have Covishield, Covaxin, and Sputnik V, which are vaccines that are in use. The Zydus Cadila vaccine has been approved for use, but is not yet available. The bulk of the supply that we have had so far has been from Covishield, but Covaxin is also increasing its production. Covaxin had planned to do studies in Brazil, but those plans have now been cancelled because there are some controversies about the approval of Covaxin in Brazil. So in terms of these vaccines, we always look at how well the vaccines are working, but we also look at safety. And with Covishield, there is a safety signal of thrombosis with thrombocytopenia syndrome, which has led to many countries saying that this vaccine should not be used in younger populations if an alternative is available. This is a low risk event, but it's important to recognize that this occurs because there is a treatment. If you can get people to hospital, if they have thrombosis with thrombocytopenia, then they can be managed easily. So recognition and treatment are really, really important. And in India, we have, a very limited supply of vaccines still. So we need to take on board the benefits of vaccines as well as the potential risks of thrombosis with thrombocytopenia syndrome. There are lots of other vaccines also being made in India. Biological E is making Corbevax, Zydus Cadila, we spoke about, Genova, Novavax, BioEase, J&J vaccine. Sputnik is looking at a single dose as Sputnik Light. Oro Vaccines, a Hyderabad-based company, is doing a VSV vector. Bharat Biotech is working on an adenovirus vaccine, which is a nasal approach. And then SII and Codit Genex and Indian Immunologicals and Griffiths University are working on live attenuated vaccines. We also have approval from DCGI for Moderna and J&J vaccines, that the indemnity discussions for those and for the Pfizer vaccine continue to be ongoing. Now, where are we today in terms of vaccination? We had the very, very high numbers of people vaccinated yesterday on the Prime Minister's birthday. So it shows us what we are capable of doing if we really want to vaccinate people and there is supply. The Indian immunization system is really very, very good in its ability to reach people. But as with any immunization program, it becomes more and more difficult to get to people once you've covered the easy part of the population, which is largely in urban areas. The more rural, the more remote you are, the harder it becomes. And we are in a situation today where about 14-15% of people have received 2 doses of vaccine, of adults have received 2 doses of vaccine. There continue to be a lot of challenges around vaccination, supply, reaching people. We are not really measuring impact of vaccines, whether we should be thinking about different approaches at different times. Lots of things that we really need to discuss about vaccines. We can't cover everything, so I'll quickly take you through the key issues. Supply, we've had a lot of stories told to us, but essentially supply has been poor, but is getting better. So will we be able to immunize all adults by the end of the year? I'm not sure we can, but we may be able to get to about 70-75% of adults receiving all vaccines. We have a very, very large immunization program, and even when we do immunization for children, we manage to reach only 90% of children. So will we be able to get much further than that for adults? I think that is something that remains to be seen. Even in 2020, which is our best program ever, we managed to reach 90%. And in the world, if we look, Nigeria and India lead the world in numbers of children that have not been immunized by routine vaccines. So even though a lot of efforts have been undertaken for vaccination, we're a big country with a very large population, and it requires extraordinary efforts to get to high coverage. The other thing is we've given out now, 750 million doses of vaccine. But the problem really is that most of the data that we have on effectiveness of these vaccines comes from relatively small studies in hospitals, studies in police personnel, the occasional breakthrough cases. We are not gathering data as a public health exercise to look at vaccine performance. And this really is a challenge. And it's a challenge because we need to think about what we want from our vaccination program. And I'm not actually clear about what we are doing beyond trying to protect people. But if you go beyond protecting people and you think about strategies, then you really need to be thinking about adaptation. Are we protecting the health system or essential services or lives or livelihoods or, future capital or spread of disease? Who decides this? I'm a member of the National Technical Group on Immunizations and of the COVID-19 Working Group for the government, and I am completely unclear about what the goals of the program really are. Now, coming to variants, if we look at variants, we've had lots of names for the variants. And maybe this WHO label, which goes alpha, beta, gamma, delta, is the best way to go. It's certainly been accepted since it was established, but it doesn't always show you the relationship between these variants, which the older numbers did in terms of lineages. Now, how are vaccines doing against variants? In our early data, it looked variable. Alpha was a variant that came out that was incredibly transmissible, but fortunately, vaccines worked very well against alpha. Beta, which came out of South Africa, had less vaccine efficacy in clinical trials and in effectiveness studies. But with Delta, we were again in a very different situation where the UK early data showed us that infection against, with the AstraZeneca vaccine, protection against infection was lower for Delta than for Alpha. And that was true for infection and for severe disease, but the protection against severe disease was better than the protection from infection. So in general, the vaccines are working reasonably well against severe disease and mortality, and variably against infection. Now, if we look at data that is variant specific, then what you can see here from England, Scotland, Israel, Canada, with the different types of vaccines, what you see here is that Delta gives you get less protection against Delta than you get against Alpha. This is much clearer when you look at all symptomatic infections. But when you look at protection from hospitalization, then the difference is a lot less. So for India, trying to understand protection against infection and hospitalization is important, but the data we have isn't always there to help us address those questions. Now, there are data that are emerging that are looking at all of the studies that have been put together, and what's shown here in the bars. First bar is alpha, second is beta, third is gamma, fourth is delta. There are studies that have focused mostly on alpha and beta variants, with many of the vaccines that came out in the developed countries. And what it shows there really is that the darker blue color, that is, you can see that the beta variant is the one where the vaccine response seems to be the least good. And this is something that has been borne out in the effectiveness studies also. So the questions that are being asked is, do we need now booster doses now that the variants have come and it appears that vaccine efficacy is waning? We know very clearly that when it comes to immunocompromised individuals, there is a reduction in the antibody response. And this is greatest for people who are solid organ transplant recipients and is relatively less, let's say, for patients who have hemodialysis. So I think when we begin to think about who needs to be getting booster doses, we need to have data that is quite granular, which allows us to understand who are the populations that need these booster doses or not. We know that when you give a vaccine, any dose of the vaccine, you will see a response. And here is a paper that shows you that you actually see a better response if you treat people who are immunocompromised with 3 doses instead of 2 doses. With 2 doses, there was about 40% of people were protected. And with 3 doses, this reached about 65%. So not like normal, but certainly better than before. In the last couple of weeks, we have had a lot of data emerging from Israel. And Israel, in fact, has started to give booster doses first to its elderly population from August onwards. And they have now expanded that to the bulk of their population. Why are they doing this? They're doing this, one, because when the Delta variant came in, they started to see an increase in cases. They started vaccination early. They covered a lot of their country very early. During June and early August, 60% of their elderly who were coming in with severe disease, 60% had been vaccinated with 2 doses. So they saw this increase in severe cases. They also saw a decline in protection from infection across all age groups. In people who were vaccinated early, they saw less protection the further out you went. And then they introduced the vaccine, and what they were able to show is that once they started giving the vaccines in the age group of 60 and over, they could reduce the amount of confirmed infection by about 11-fold. So in Israel, they reduced infection, they reduced severe disease by giving booster doses to a population that already had very high levels of infection. We also have data that has just come out from the UK on severe disease with the Astra and the Pfizer vaccines. And if you look at these graphs, it looks like a decline. But what I'd like you to focus on is that the decline is to 80% protection against hospitalization. And if you recall, what we were looking at from the WHO was at a vaccine being better than 50%. So what is the level of protection we want to achieve? What is the strategy with which we want to achieve it? What does this mean for India? I think this raises a lot of questions. In India, we are not the same as UK and Israel. We are a country that is 2/3 seropositive, with the bulk of that positivity being acquired through infection. So if that is the case, and we know that infection and vaccination gives better protection than vaccination alone, do we need to boost? And if we need to boost, which is the best booster? So at the moment, I don't have clear answers. I have thoughts, but I think we need data before we make decisions. And one of those, it relates to which vaccine we should use for booster doses, or whether we should think about changing our primary immunization approach. And there are a number of studies that are being undertaken around the world that are looking at immunogenicity using a mix and match approach. There are a few reports that are available already for the mRNA and the AstraZeneca vaccine, but there are a lot of other platforms around the world that also need to be evaluated. Other questions relate to dosing schedules. This has been of particular interest in Kerala, but I will tell you that the longer the gap we have between doses, the better the immune response winds up being. And there are data now to show that in multiple locations in the world, including in India and in Kerala. So there are many other public health questions that are important. There are lots of things that WHO wants us to think about. Vaccine hesitancy, slow vaccine uptake, programmatic issues, whether there's funding, the fact that we are not reporting safety events very easily, the fact that we don't have clarity on when supplies are coming. All of this creates complications. But ultimately, I think what we have to remember is that vaccines are an absolutely outstanding tool in our ability to control SARS-CoV-2. And it is control and not elimination that we should be thinking about now. So with that, thank you very much for your attention. And for the remaining few minutes, I'd be happy to take questions.
Thank you so much, madam. Your years of clinical research and your experience in this field is very evident in your talk. And I'm sure the students especially would have benefited a lot because you have walked them through the basics of the morphology relevant to vaccines, the immunological aspects, and then the types of vaccines, and you've formed a base so that they can build upon. I'm sure there are a lot of questions in the chat box. Madam, shall I read the first question?
Sure.
Okay. The first one is from Dr. Tina. She has asked, does COVID vaccine have cross protections to variants? I think madam has already answered that now.
Yes, so the figure that I showed actually has much more data on it. It's a review that is available in Nature Reviews in Genetics. It came out, I think, yesterday. So you might want to look at that. But yes, there appears to be the greatest reduction in protection and backed up by clinical data against the beta variant and then against the gamma variant, then delta, then alpha. So delta is, fortunately for us, vaccines are working quite well against the delta variant.
Thank you, ma'am. And the next question is, the interval between doses of Covishield vaccine is constantly changing. Why?
I think, from an immunological perspective, the best interval between vaccine doses is 4 to 6 months. But usually we don't wait 4 to 6 months. So here what was done was an attempt to get the maximum value out of the vaccines by ensuring that we had a longer interval. I think that, actually we started with too short an interval and we should have expanded it earlier. A lot of people have objected to it, but I think one of the things that is important to remember, we blame the government saying there wasn't supply. But even if there was supply, the longer interval is a better strategy, immunologically.
Thank you, ma'am. The next question is, how do we educate people who approach us with clear cutoff ratio above 1 and interpret it as a protective level?
First, tell them they're measuring the wrong antibody.
Yes.
Most of the CLIA tests, well, all of the CLIA tests that we have are measuring binding antibody and not neutralizing antibody.
Okay, ma'am. The next one, is it worthwhile doing antibody levels from any commercial source after getting the vaccine? Do we need to be worried if the levels are low and undetectable? Similar lines, I guess.
Yeah, so I think one thing that is important to remember is that for no other vaccine do we bother to get antibody levels. Occasionally in healthcare workers, we might say, okay, look at your hepatitis B antibody levels, but occasionally. Don't usually do it for any other vaccine. Why would we want to do it for this vaccine? Now, if your levels are low or undetectable, yes, it's likely that your neutralizing antibody levels will also be low. Do you need a booster? We don't know that yet because we don't have a cutoff. So mostly what I've heard is that for many of the tests that we are seeing and doing, the test has a very low dynamic range at the bottom of the distribution. So there may be antibodies, but we are not measuring them. And from at least the ChAdOx studies, it's been shown that very low levels of people with very low levels of antibody were also protected.
Okay, ma'am. The next is, what is the ideal interval for a post-COVID patient to take the first dose of COVID vaccine?
Ideally, 4 to 6 months. But, what is recommended by the government is 3 months. But, if you've already had COVID and you're going for vaccine, you're actually going to be in the best possible position for protection. So not that I advise anyone to go and get COVID, but if you have had it and then you are vaccinated, or you get it after 1 dose of vaccine, it really appears that the combination of natural infection and vaccination gives the best protection.
Ma'am, next question is there any robust testing available to check neutralizing antibodies other than by doing neutralization tests?
Not at the moment. There is a test which is the GeneScript assay, which is a surrogate neutralization test, which looks at the ability of antibodies in serum to inhibit binding of the spike protein to ACE2. But that also looks at a relative reduction. And it's a reasonable test, but it's not a perfect correlative yet.
Okay. Next is, do we have data suggesting Covaxin is better vaccine than Covishield?
No.
I guess, again, data is still not available. So
yeah, we have some effectiveness data now for Covishield. We have no effectiveness data for Covaxin.
And which vaccine do you think is safer for young individuals, especially in regards to side effect profiles like TTP?
So I think with all of the side effects are very rare. TTP is very rare, and TTP that can be recognized and managed is something that is easily handleable. The question is, are we educating people so that they can recognize it before people wind up in hospital? So with the mRNA vaccines, we have viral myocarditis, which is much higher in young males. We have TTP. Inactivated vaccines usually have the least side effects, but inactivated vaccines also have usually the lowest effectiveness. So it really is a question of what are the benefits and what are the risks with each vaccine. In general, these are very, very safe vaccines. One of the side effects that's not discovered that seems to be across many vaccine platforms is zoster. So lots of people are reporting reactivation after receiving doses of the vaccine. It's been seen with AstraZeneca, it's been seen with the mRNA vaccines, and no one has a clear understanding of why.
Next question is, ma'am, do you anticipate any new variant more lethal and transmissible than Delta?
Yes. I think as long as the virus continues to replicate, there is a danger that we will see new variants. And this is a virus that acquired its initial mutations through mutation. But in RNA viruses, we also have the possibility of recombination. So if there is a mixed infection, two viruses can trade chunks of their genome. And if you get recombination, you'll wind up with a string of genes being transferred. So it's possible that we can wind up with something that is more transmissible, more virulent, and capable of the immune response. All of these are low probability events, but they are not impossible.
Another question is it because of variations in the immune system of different individuals that is making vaccinated individuals infected?
Well, to some extent, yes. You will make different levels of antibody from me. And if antibodies are a correlate of protection, then it is our response that determines our protection. The vaccine is not different. What is given to us is the same. And the same goes for response to a natural infection also. Why do some people have asymptomatic infection and others develop severe disease?
And can mixing COVID vaccines give a better immunity than booster doses?
In terms of immune response, that seems to be the case from the studies that have been done. But will that translate to better efficacy? We don't yet know. And I'm not sure that we will know for a while. But if we use immune response, you may wind up with more robust response with a mixed approach.
I think questions are keeping on coming in and everyone wants you as the question answer bank. So,
but
it's
I
already...
think you need to move on. Yeah. What I'll do is I'll stay on for a while and I'll answer the questions in the chat box. Okay.
Thank you so much, ma'am. So once again, thank you for this engaging lecture and question answer session. Next, we'll be moving on with the presentation. I share my screen to just give a brief introduction about Dr. Rajiv, sir. So next we have with us a prolific speaker. Sir is very well known in our circles in Kerala and especially even in Tamil Nadu and down south. And actually sir is started as a big person itself because from Kerala state he started with second rank holder in SSLC, went on to first rank in medical entrance, got first in MBBS and CMC and gold medal in MD General Medicine. And not just he stopped with that, he went on to the UK, got MRCP, and sir has passed the fellowship exams in America for medicine and gastroenterology. So sir is very interested in epidemiological aspects and sir has written over 70 more health related articles. And sir's Twitter account is one which if you have not started following, follow. So please, that is a rich source of latest scientific developments, which is updated very regularly. And sir has written a book, Think Like a Doctor. And sir currently holds many positions in the Kerala State IMA Research Cell, Epidemic Control Cell. And sir is also the convener of the Communication Skills Training in Kerala State and the Scientific Committee and past president of IMA Cochin. So now over to you, sir, to give us an enlightening talk on the public health aspects of COVID-19 via 19.
Thank you. And the topic given to me is to discuss the epidemiology of Kerala. The questions that I commonly get in this context are, why is Kerala having a problem while the rest of the country is not? I'm sure many of you in the audience have seen or read multiple points for and against this argument. So I'll explain to the best of my knowledge what is happening there, and then I'll go on to discuss some special points about how we manage the pandemic in Kerala. When I say we, I mean the entire Kerala state, including every single citizen here. So before we discuss what is happening in Kerala, I'd like you to take you to two countries. That is Sri Lanka, that is right next door to us, and also to Vietnam. So now Vietnam is considered the nation that had literally conquered the pandemic with fairly limited resources. They are not exactly a super rich nation, but they did all of their public health measures right, and there was very little disease in 2020. When the rest of the world was struggling with severe tragedies, Vietnam, there was really no disease at all. We know that for sure, and that was reflected in the zero surveys that in January of this year, can you believe it or not, only about 0.8% of people had antibodies. Now, that's incredibly low because that included healthcare workers. Can you imagine a healthcare worker having 0.8% zero prevalence? And that showed the success of the shielding. And now you fast forward that to April, May, and in comes Delta variant. As I mentioned earlier, it's like Delta variant is like Godzilla 2. It's huge. It's huge. It's 1,000 times more powerful, bigger, meaner, spreads faster. And if 2 people were infected in a home last year, now it's 4 people and so on. So we all know about Delta. I'm not going to go into that. But why did Vietnam have this huge surge? Now Vietnam is having a massive surge, prolonged lockdown. Why is that? Now let's come back to our neighbor, that is Sri Lanka. Sri Lanka did very well. They engaged the military very early, and the military and the health system worked hand in hand to literally control the pandemic. And they built, almost like a containment zone. They built isolation facilities, and everything was handled like clockwork. And they did very well. And their zero prevalence at the end of the year was about 22%. That's not much. And Kerala, by the end of the year, with all our measures here, we hit just about 11% by December, just 11%. So which meant that by the turn of the year, almost 90% of people who live here were completely unexposed to the virus. So then when Godzilla 2 came to Kerala, Godzilla found that Kerala was the perfect place for it to work. But fortunately, by decision, the state locked down just when Delta hit. I think we may have been a few days late, but it's very difficult to time these things, but the state locked down just at the moment of crisis. That's the only time Kerala just about broke a sweat. I could see the stress in healthcare workers. So we locked down and we kept Delta out. And when we opened up, Delta was still around. It didn't go anywhere. It was silently spreading in the homes. How? Because unlike last year, it is able to spread to many people more quickly than it did last year. And so Delta hadn't gone anywhere when the lockdown was lifted. So Delta continued to spread, but not so much. If you see the post-lockdown curve in Kerala, I know the cases really went up to around 20,000. They went up to 30,000, but they really did not break the ceiling. They did not. And as of today, the cases are hovering around the 20,000 mark. And what is more important is our hospital system, our healthcare system has never been overwhelmed since the surge. Obviously, when we locked down, clearly there was some strain, but nobody was denied a bed and nobody died on the street. The strain was more felt by our healthcare workers who had a tough time during the surge. So that is where we stand right now. So that is my answer to the question on why Kerala. And now many people lay people have a binary view of many things. They see things in black or white, good or bad, etc. And you'll see that a lot in articles written about the pandemic. The pandemic is a 3-dimensional subject. And the spread of a virus itself, as this audience knows very well, is multidimensional. And I'll take you to what is called the epidemiological triangle, which is common for all infectious diseases, for your information. So infectious diseases can be in bacteria, viral, protozoal, whatever. So the 3 parts of the triangle are agent, host, and environment. Now, agent is the virus in this case, host is human beings in this case, and environment is what separates the agent and the host. So let me take you through another example, cholera. See, cholera was a devastating illness that killed many, many people in the past century. But Once the source of cholera was identified, that hand pump in London, the doctor who discovered the hand pump, the doctor found that anyone who drank water from that hand pump got cholera. And that was a turning point in the history of cholera. It is not like we developed antibiotics or vaccines for cholera. We were able to conquer its environment. Now, environment is such a powerful phenomenon. And for cholera, it is actually water. So we gained control on water, and that is how cholera was defeated, not through vaccines or antibiotics. Now let's switch back to COVID-19. COVID-19 is caused by a virus which almost exclusively spreads through the environment, that is air, not on a surface. See, my table is not going to have a lot of SARS-CoV-2 virus, but we spend an enormous amount of time, money, and effort and stress in cleaning all these surfaces for what? So there is hardly a single case report of SARS-CoV-2 virus transmission from an unclean surface, but still we do it. I'm not implying that we should not clean surfaces, but I think these resources should have been better used in controlling air. And when I say air, I'm talking about multiple factors that involve the air. The factors that, such as temperature and humidity, are beyond our control to a large extent, but factors such as ventilation are within our control, very much within our control. With very minor changes, we will be able to change the ventilation or air exchange rate of the environment we are in. For example, say you're working in an office, if you open your windows on both sides of the room, so the air comes in on one side and exits on the other, yes, that's good ventilation. Whereas if you only open one window, it's not going to provide much air exchange. So these are ways in which we need to move forward, we need to think. And whenever I discuss these topics, which are obviously based on scientific articles that are written on it, I get dumb looks. People look at me as though this is something which is completely wrong or impossible, and etc. Let me ask you a question. When the pandemic broke in early 2020, if someone said that the majority of Kerala will be infected and a large number of people will die, and that all of us will be wearing masks for a long time, do you think anyone would have thought he was sane? No. So, which is why we are facing an unfamiliar enemy, which is powerful, and it's going to be here for a long time. We need to think forward, and we need to look at ways to outsmart the enemy and not just rely on vaccines. So that is one of my main messages for the audience here. Do not just think vaccines are the way forward. There are multiple other ways in which we can fight this pandemic. And I profoundly disagree with the statement, let's live with the virus. That's completely, totally wrong. We are not going to allow this virus to take control of our lives. And we need to invent and conduct ourselves in a way that we will continue to fight this virus until it is either extinct or we get a very efficient antiviral agent, which is yet to be invented. So those are some thoughts about the environment. And then I'll take you through why Kerala did not really have so many deaths. See, this is deaths in the pandemic is a very interesting medical topic to study. Death is tragic, but I'm talking here from an academic standpoint. If you look across the world, all countries, particularly the developed and the richest nations, have all gone through surges of deaths. And if you examine closely why that happened, it is when the healthcare systems were overwhelmed. Anytime your ICU capacity or your oxygen bed capacity they don't have to overflow, but if they're near capacity itself, the care and the amount of care given comes down. So that is the main driver of mortality in COVID-19. So if you look at Kerala, for example, so you might wonder, how can a rich country have COVID deaths? As I mentioned, you could have a ultra fine ICU bed with the finest nurse in the world and the greatest critical care specialist standing beside him or her. But if you have 10 patients who are equally deserving for that single bed, you know what? 9 will probably succumb or have morbid outcomes. So that's the point here. So we need to either make more ICU beds or reduce the number of patients coming into the ICU bed. So that is really where Kerala stood out. Kerala's capacity was enhanced significantly, but what we really did was something very impressive that's not been talked about much. I'll explain how it works. Now, I want you to imagine a Christmas tree that looks like this. It's a tall Christmas tree with a single stem. And as you know, the Christmas tree is broad at the base and very, very narrow at the top with just a very, I'm talking about a traditional Christmas tree. Now, imagine that the top of the Christmas tree is the ICU, or the ICU in the apex centers in, let's say, in a state or a country. Now, let's imagine that the pandemic is hitting this country and everybody is rushing to the apex center. So everybody who's got a fever rushes to the apex centers, and the apex center is suddenly full. See, this tree is going to get heavy at the top. See, all the people are going to run to the top, and that tree is going to bend forward and crash onto the ground because it becomes top heavy. The lower branches that I'm talking about are actually the primary and secondary healthcare systems and the primary care clinics and the individual doctor clinics that have been actually the backbone of healthcare in Kerala. Kerala's health is not maintained by large tertiary institutions. Kerala's substantially good and much improved public health is largely maintained by individual doctors who run clinics and by small healthcare establishments, including primary health centers and secondary health centers, not by tertiary. So my point is that there's a large number of COVID-19 patients that can be treated effectively by these grassroots level establishments. And that's really what we focused on from Indian Medical Association, from the Kerala Government Medical Association, Officers Association, KGMOA, the Kerala Health Services. What we did from very early on, from January onwards, was to empower the doctors and the nurses to handle the pandemic at their level, rather than refer the patient to the topmost branch of the tree. And that really is Kerala's success. So if you saw the apex center, they were almost never overwhelmed. There are focus exceptions once in a while. Some centers might be full for some time, but there are always other hospitals nearby that can take up the slack. So that's really what Kerala did. We did a lot of work in empowering doctors. Indian Medical Association did literally several seminars every day for months together. We are still doing it. And we are educating doctors on what the latest treatments are, what treatments do not work. That's very important. There are many treatments that were empirical, imaginary, and not evidence-based that have all been thrown out of the window. And our doctor fraternity has really stood with us throughout, and their confidence levels has gone up. Not only did we empower the doctors to treat COVID-19, we also taught the healthcare system or healthcare workers to protect themselves from dying. We had put out the earliest publications on healthcare workers' deaths from around the globe. We also published on the first time anyone published on healthcare worker deaths was from our unit in Cochin. The first survey on post-vaccination symptoms was from our unit in Cochin. So we were able to impart substantial learning on all levels of healthcare providers, including our nurses. Earliest article on when to protect nurses, how to protect nurses, went out in early April. And that was severely frowned upon, because that article opened with a line that said, imagine that everybody around you has coronavirus and then treat the patient. And that statement still holds true to the day. And I wrote that statement, and I wrote it in April of last year. But it has been well received, and our nurses have not died. In our state of Kerala, I think tragically we have around 20 or 22 deaths among doctors. While that is an enormous number for anyone, that number is minuscule compared to the rest of the world. So Kerala has done a few things right. And that is, I believe, by repeated, relentless, untiring efforts by the doctors and the nurses and the administration here, with a huge support that we received from the media. See, doctors can have all the scientific knowledge and expertise that they want, but if that knowledge does not percolate to the grassroots level, it is of no use to anyone. And we've seen that happen to some wealthy nations without naming them. There are smart people there too, but their knowledge never reached where it is supposed to, which is the grassroots level. Here in Kerala, you ask any ASHA worker or any citizen in Kerala, they will tell you the basics of the pandemic. Some of them may not be following it, which is quite fine, but that is expected. But the vast majority of people already being educated by Nipah, 2 bouts of Nipah, they were ready for the pandemic. But unfortunately, as the pandemic wore on, fatigue crept in, and people got tired of doing the same thing, and people were tired of losing their income. Remember that it is okay for a healthcare worker to draw a salary, and because this audience is primarily healthcare workers, but for the average person who makes a living from daily wages or daily trade, these past 2 years have been really, really difficult for them. And I know families who have told me that they would have died from starvation had the government not given them rice and beans from the ration shop. I know this for sure. And while we make fun of governments while governments get trolled for all this, I know that these things are profoundly important, because if you are a daily wage person and you know only one job, and if your job is not there, how will you feed your family? I think that is an extremely important aspect of the pandemic that I think Kerala has really addressed. So we did not really have that many tragedies, as it could have been a lot worse. To mention a few adverse events, the light and sound industry in Kerala, I'm just singling out just one industry. I work on noise pollution as well. I work on multiple public health fronts. Pandemic is just one of those. So the light and sound industry, we work closely with them because we sat with them for control of noise pollution. And Kerala is one of the states that actually has done giant strides in noise pollution. But back to this light and sound industry, they literally went jobless when the pandemic hit. The lockdown affected them so severely. This whole organization of, I think they have several thousands of members in that organization. And they were all in severe distress. In fact, multiple suicides have occurred in that segment. And so these are things that we should not forget. Sometimes we get carried away a little bit by vaccines and fancy monoclonal antibodies, which cost upwards of $1 lakh. You know what? The control of the pandemic is really at the grassroots level. I think we must never forget that. Among all the tools that Kerala used in controlling the pandemic, I believe that the area that I specialize in was the most important. I am the state convener for communication skills for doctors for the past 8 years. Some of you have probably seen my seminars or attended them. They are a continuing learning process, and I think Kerala used effective communication to connect with the grassroots level. Not just the doctors, but every evening the chief minister actually talks directly to the people, and that is extremely important. I know this can be trolled on a political ground, but I'm not a politician. I'm talking about science here. If the people have one spokesperson they listen to, they will tend to believe them all the time. And to me, honestly, it doesn't matter if it's one person or another person, as long as the same person doing it and doing it effectively, consistently, at the same time, rebuking them at times for not following instructions, and updating them to within our limitations. Kerala's case finding has been exemplary, as all of you know. Just to explain what case finding is, suppose we have an unknown village, let's say a hypothetical village, and there are 100 cases of COVID-19. How many cases can that village detect is called the case finding system. If the case finding system is very robust, the village can detect all 100 of them, which means the case finding ability is 1 out of 1, which is great. That no system is so perfect, because the pandemic itself comes in multiple shades, from white all the way through black. In other words, you can have completely asymptomatic people walking around as though nothing's wrong with them, and then you have people who are severely ill on a ventilator. So back to this village, if the case finding system is not very good, then the system finds only about, say, 10 out of 100 cases, then the finding ability is 1 in 10. The case finding system is worse. It picks only about, say, 2 out of 100, then it's 1 in 50. So we call the case finding system is 1 in 50. Kerala's case finding system is so good, it rates at 1 out of 6. We are able to pick up 1 out of 6 cases that are actually happening. You know, India's average rate for a nation, it is 1 in 33. India, the average case finding rate is 1 in 33. And there are some states that because of certain limitations of infrastructure, their case finding ability is beyond 1 in 100. So Kerala's case finding ability is good. The advantage of a case finding ability is twofold. One, you can communicate effectively to the public that these are our findings. And secondly, if you identify a case, you can isolate them and you can isolate their contacts. So both ways, the case finding ability is extremely important. So that is another aspect I wanted to highlight. We were also quite aggressive. I'm talking about healthcare fraternity. See, you can't just sit back and let somebody else do the work for you. You have to be in the forefront. Many of us have been in the forefront 24/7 ever since last January. And I'll give you a couple of instances. Whenever fake news surfaces, fake news is like weeds. They'll always grow in your garden and you got to go and pick them out. If you let weeds grow, they'll just overpower your garden and your garden will not exist. And fake news works like that. Places like US are flooded with fake news. South America, Europe is flooded with fake news. In fact, 30% of Americans are refusing to take the vaccine because they think there's something wrong with it. Such is the influence of fake news. But these sort of characters hang around in Kerala as well. But whenever they've come out with their next trick, we went on the offensive so hard. We went so hard at them. We quelled every single attempt based on merit. If it's a lone person who's just making a little bit of noise, we wouldn't give them the publicity. But if it is a major viral fake news, I personally took it up. Many of us doctors, we went, Dr. Sulfi, so many of us, I don't want to name them. There were so many of them to name. We all went on the offensive. We went on television, radio, WhatsApp, Facebook, Twitter, whatever, on the newspaper, digital media. We were on every medium saying that these things are wrong. And of course, whatever you do, you get criticized, and that's the Kerala society, which is fine. But in the end of it, fake news we never allowed weeds to grow in our garden. So I think the latest fake news that came out was a viral voice clip about deaths occurring after tetanus toxoid and COVID vaccine. We went on the offensive so hard, the news just died in 2 days. So my point is, unless you are proactive, you cannot win. So these are ways we did it. And another example I'll quote here, we got a secret information that an anti-science group was going to demonstrate against wearing masks outside of Kerala High Court. This occurred last year. We got whispers about it. We immediately contacted the authorities and we had the person arrested. Yeah, the person was arrested and reprimanded. So that's how you fight pandemics. You can't just sit around and wait for things to happen. So that is, I'm just giving you a feel for the intensity of work that we did. One of the things that we did from at least our unit in Cochin was from March onwards, under the leadership of many doctors. I don't want to name them. There are too many doctors who are involved. We set up a massive control room, which basically was a common desk, common confluence of public health system, government services, private doctors, volunteers. Everybody came together on the same platform. We set up information broadcasting systems. We set up patient transport systems. When the lockdown occurred, we delivered medicines to their homes. We had help desks, which were coordinated through the district administration. We had exclusive helplines for doctors who were undergoing stress during the pandemic. We had helplines exclusively for doctors who had scientific doubts about the pandemic. Again, very early. Remember, these were very early days when people didn't know much about what was going on. We had later in the pandemic, the unit distributed PPE kits at a very discounted rates. We mobilized funds from CSR funds from multiple organizations, converted that into PPE and had it delivered to frontline workers. And intense work has occurred. At least I'm just giving you a sample of the work that was done over here. And similar work has been done from all districts, I'm sure. Then we also had regular brainstorming sessions with doctors. Our Tuesday meetings are so famous, and it's the talk of the town. Ever since the pandemic started, we ran Tuesday meetings, 8 p.m. A bunch of us will get together on Zoom and discuss, like friends, what exactly is happening around. So each of us have a different exposure, experience, and background. Some of us are frontline workers, some are in research, some are in administration, some are with government, some are with public, but we all sit around and talk. And you know what? Conversation helps. So we've warned the policymakers, we've sent advisories to the government, we've sent advisories to the public, advisories to the media. So it is a collective effort that actually has gone behind the Kerala's control of the pandemic. So my point is that there is no single way of, there's no single person who can take credit or single methodology here, but the pandemic is a subject that encompasses around 15 different subjects. That is, you got your virology, you got your immunology, you got ethics happening here, you got communication happening here, physics plays a major role, engineering plays a major role, pharmacology does a huge role, physiology, we need to be very strong in physiology, pulmonology and critical care extremely important. Genetics, if you don't know your genetics, then you're going to be stumped by every news that comes at you. You got to have your biostatistics so strong that you should be able to tell a fake literature from or a badly done study. So the pandemic encompasses multiple different topics. So to the people who are interested in the academic part of it, I would encourage them to read up multiple subjects so that they can form a very firm base. We also unfortunately had a few fake messaging from some medical doctors as well, who gave out wrong messages. So, and we had to stand up and counter them as well. And these were people of substantial influence on the public who gave them wrong messages. And fortunately, we countered them. And I think this occurred last year as well, with all due respect to the people who gave these messages. So the Kerala graphs, I think all of you have seen the Kerala graphs. We've been substantially different from the rest of India. I'll take you through the Kerala graph from last year. Kerala graph never really took off until October last year. When the rest of India was in crisis, Kerala was hardly even seeing cases here. And that's because of good shielding, as I mentioned at the beginning of my talk. But by October, the cases had gone up, and I think we had reached one peak in October, and then it came down. We had another peak in January that we picked up from here in Cochin. We alerted the government. In fact, it came on the national media, state media, and the government took proactive steps. It came down. And then came Delta. So after Delta, everybody knows what happened. Now, I'm now around half an hour into my talk. I'll talk for 10 more minutes on multiple angles of how Kerala has been handling it. Because I've been asked to talk exclusively about Kerala, but I think I will spend about 10 minutes explaining some of the latest literature that has come as well. I think that's important in this discussion. Dr. Kang actually mentioned a few research studies. I'll take you through a few more which are of relevance here. I can't explain all the studies. I can't discuss all the studies here, but I'll quickly run through some major trials that have actually occurred from Kerala. Studies that came out of Kerala. I mentioned a couple from our end, so I won't repeat those. We were the ones to say that if you take a vaccine, you're not going to die. And that was very important messaging for the public. We were the healthcare workers who took the vaccine. We put a study out of 5,396 healthcare workers who took the vaccine and gave them beautiful graphs that showed all these symptoms will go away in 24, 48 hours. And so that was greatly reassuring to the public. So it is important to go out there and tell them, look, this is our experience, and none of us died. And out of 5,396 people, nobody had any major side effect. And that's a very powerful early message that we got out of the community. And that, in fact, I think laid a foundation for the lowest vaccine hesitancy perhaps in the whole world occurs in Kerala, because people want the vaccine. There are a few people always who will oppose the vaccine, and those numbers will always be there, but we should keep them to a minimum and make sure that they don't propagate the views the way it is happening in the Western media. There was one study out of KIMS under the leadership of Dr. Rajalakshmi and Dr. Mohammad Niaz, both supremely brilliant academicians who contributed a lot of papers. Look them up, Dr. Rajalakshmi and Dr. Niaz. They sit in our meetings as well. KIMS produced a study that showed that 8.2% of healthcare workers were getting breakthrough infections. And this was correlated with Vellore's, CMC Vellore's first study of 8,991 healthcare workers. And Vellore, which is my alma mater, and Dr. Kang's alma mater also, they showed that about 11% of healthcare workers were getting breakthrough infection. Now, these were the earliest data that your vaccines are not like your measles vaccines or polio vaccines. You take your vaccines, you could still get infected. And that is the fact that we communicated very early. And it's because we communicated early that people stood with us. Remember, one of the problems with vaccine communication has been lack of transparency. If you start trying to fool people into saying that vaccines are completely faultless and vaccines can never harm and vaccines will always protect you and so on, I think that kind of messaging has occurred mainly from the Western literature. That's completely wrong. I think we must be transparent. And if there's a problem, I think we must stand up and say there's a problem, and we've actually done that. And by doing that, nobody has gone against vaccines here. So that shows that if you're transparent and honest and admit the limitations, I think that's extremely important. I know there's plenty of differences of opinion. There are some doctors who believe that no side effects should ever be communicated to the public. I think that I profoundly disagree with that. I think if you say it with sincerity and with goodwill and good intent, I think the public will absorb it and they will say, okay, I think we will go ahead and take the vaccine. Some other studies that came out that merit a couple, I'll just take you through a couple of studies. One study is very interesting. I have around 7,500 tweets, so you're welcome to check that. Some tweets take me about 3 hours to do. It may be 3 sentences, but those 3 sentences take me about 3 hours to do, because to digest some papers and verify certain things, it takes me time. So it's a service to the science. So out of those, I've selected a couple of studies I thought I'll discuss in the next 5 minutes that I have here. One is a study on monkeys. You know, there's a study that came out of the West of 16 monkeys, a beautiful study. What they did was they exposed these 16 monkeys to increasing dose of virus in the air. You know, monkeys were made to inhale virus in increasing doses. So monkey 1 got the lowest dose and monkey 16 got the highest dose. And guess what they found? I'll give you 2 options. Option A, all monkeys got the same disease, which is like your all or none phenomenon, which is what I sort of believed in, because there's something called the transmission bottleneck of this virus, which basically means that if you, no matter where you get the infection from, ultimately the number of viruses that infect you physically at the start is less than 10. And that's called a transmission bottleneck. You might want to look that up for those of you who are interested. But back to the study, they found, if my theory was correct, all the monkeys should have got the same disease. They did not. They found that the monkeys, the 6 monkeys who got the highest dose, developed symptoms, that is fever, and also seroconverted. They developed antibodies. So the 6 monkeys at the top, that's the highest dose, got seroconversion and fever. The 6 monkeys below them, that's lower doses, the middle doses, they did not have a fever, but they seroconverted. Those were the asymptomatics. And then there were, I had 6 and 6, so that's about a few monkeys, about 6 or so, 5 or 6. They did neither. They didn't have a fever and they didn't seroconvert. And therein lies a most interesting aspect. And one of these monkeys that neither seroconverted nor had a fever had virus in the nose. So this is perhaps one of the finest studies in the world on the pandemic. I want you to look it up, because this is done by eminent researchers. And what they found is there were actually 2 monkeys that had virus in the nose. One monkey had virus in the nose within a couple of days of getting exposed to the virus, which is maybe dead virus from inhalation. But the monkey that actually had virus in the nose, who had no fever and no antibodies, had in fact shown us that it is possible to have an exclusively nasal infection from this virus. And this speaks volumes of how this pandemic has propagated throughout the world. Sometimes the smaller studies give you the best detail. It's a lot more to talk. There is time constraint, so I'll stop now. I'll take questions for the next 15 minutes. And if there are no questions, I'll keep adding some topics and go on till 3 o'clock. That's my allotted time.
Thank you so much, sir, for that superb presentation. Sir, you have a wonderful gift of holding our attention sentence by sentence without any help of a PPT. I think that's remarkable. And on behalf of everyone gathered here, I thank you. I think the main question everybody wanted to ask, you have already answered in your talk. So why Kerala? There's been a lot of controversy of Kerala being in the 4 lines. We are illiterate people, but then still you all are having a lot of cases. So you have gone through all those aspects. You have given the analogy of the Christmas tree, how our system works here, and you have told about how to be proactive in the public health care and how you have to keep on doing things rather than just watching from the sidelines. So it speaks volumes to us. Thank you, sir, for this presentation. Any questions? Participants are also free to directly unmute and ask. So that's also there if you want to directly ask, sir. Or sir, can you can
last year I think people may be afraid to ask questions directly, I think last year, remember, as Dr. Kang said, we were helpless. We had A, no vaccine, B, we didn't know that a vaccine would ever work, and C, we were trying all sorts of strange, what we call repurposed medicines, which is there are less polite terms for that, so I don't want to go into that. Such medicines were used, but look at where we are now. We have vaccines that work. We know that they reduce deaths, and in spite of all the 20,000 cases that are occurring every day, our systems are not overwhelmed. The ICUs are not full. Death rates are not going through the roof. So I'll say this, that if you look at the graphs of countries that are well vaccinated, you can see the graph of cases and deaths are diverging. I know that's a lot to take in. I'll explain once more. There are two lines in a pandemic: the graph of cases and the graph of deaths. So all of 2020, these graphs went together in all countries. But this year, if you go and search country by country, you'll see that obviously coronavirus is going to come in waves all the time. Multiple waves are going to come. But the question is, these waves are happening, but the deaths are not going up. So the cases will go up, but the deaths are not proportionately going up. And if you see the UK graph now, who used the AstraZeneca and Pfizer vaccine, their deaths are not going up. They are not. Unfortunately, in the US, the deaths are going up, and I'd be keen to know how this actually progresses. Did they use? Was it a function of the vaccine? We don't know. So it's too early to say. But I think come next year, we may have some surprising findings about some vaccines being better than the others. I don't know. These are early signals, and Kerala's failure to increase severe cases is a classic example of how we've started conquering the disease. Given that we have very low seroprevalence compared to the rest of the country, our vaccination rate is very good, and our cases are not going up compared to the same period last year. So vaccines have provided us with a golden opportunity. And Dr. Kang has already discussed about vaccines, so I don't want to repeat anything that she has said. I think the way forward is ventilation. We really have to get this into people's heads to improve ventilation systems. Now, ventilation systems for a closed room are complicated. It's not my area of expertise. I know about it, but I'm not an expert. You need to bring in the experts. Where are the experts? I don't see them on any television panel here. I see the same, people who are politicians talking about RT-PCR and TPR, which is completely useless. Why would you want a politician talking about TPR and RT-PCR, which they know nothing about? These are complex medical topics that they are talking about in very superficial terms. Unless you have a background of healthcare, it's very difficult to understand even what that means. So I think we should bring in the experts on ventilation, have them in the forefront, have them sit in policy-making decisions. And if you are in a room, I'll take you through the basics of air exchange. See, just like my weight is measured in kilograms, air in a room is measured in cubic feet. Now the question is, how many cubic feet in a room are being replaced by outside air per hour. That's called air changes per hour. It is really not rocket science. It's like 2 buckets. So if you change the water in a bucket X number of times in an hour, then that is a number of changes per hour. I think that concept needs to come in. Air changes per hour can range anywhere from 8 to 15, which is ideal for different kind of settings. For example, if you're a bedroom that is only one person is using or 2 people are using, an air change rate of 8 is fine. If the room air gets replaced 8 times in an hour, in a flight, the systems are so good that the air gets replaced by the outside air so many times an hour. That is why flights are safe. It's not because you're wearing a mask. It's because they're taking out the aerosols that people are breathing out, which is also why there are a few outbreaks reported in airplane toilets, because they don't really have that much of space, and aerosols tend to get through to the person. See, if you have an air conditioning system, HVAC, they're called HVAC systems. They all have different gauges, different grades of air change rate. We need to start working on it now. We need to make our schools safer. When we talk about schools, we talk about opening or closing. It's not about opening or closing. It's about how safe is your classroom. Is your classroom well ventilated? Is the staff room well ventilated? Does the toilet have a basic exhaust fan? I've personally trained almost 1,500 school principals, including 150 people yesterday. And I've sat through them pretty much like this and have taken questions from them, and I've trained them personally on the basics of the pandemic. And they've all been immensely grateful to be told the truths about the pandemic and what they can do for the students. So I think there are things that we should do, and not just sit back and look at some numbers that appear on your television screen every day. All right. Any other thoughts, questions coming up, or we can stop now?
There was a question on schools which you answered before I asked you. Sir, one question regarding the role as a doctor in curtailing this pandemic. I'm sure everybody knows that we all play a major role in the healthcare system, but as an administrative support or a person going into the the state government or the central government, how can we change their outlook?
See, I'll tell you something. You asked my advice. This is my advice. No one's going to come to you and ask you for advice. That's for sure. Nobody wants any advice because there's so much advice around. So that's fact number one. Fact number two is, if you speak in measured tones, with substance repeatedly, you will be heard. And that's my experience. But if you just want to make a fiasco about something, then you may rise to fame for a couple of weeks and then you'll fade more rapidly than you rose. And I've seen that. So my point is, dogged work, sincere work. You have to be out there in whatever area that you specialize in. It may be communication, it may be some other sort of work. I think you have to, this is a special time of the century that we are going through, and all of us have to do our bit, and we all have different talents. We just have to do something that is out of the ordinary than just focus on our work. And sometimes routine work itself is good enough. Not to, that can happen because there are some people who do very, very extended hours. But I'm saying that there are other things that we can do. For example, we can influence society through social media. We can give out short videos. We can talk in our family groups. There are many family groups that are full of such junk that people are even afraid to open those messages. We can get into those messages and say, look, this is wrong. I know doctors are reluctant to do that because they'll get yelled at. But the point is, we should go out there and be prepared to take some flack. And if you're afraid to take criticism, which many doctors are, I'll tell you a couple of tips. If you're on social media, you'll be trolled, no doubt. Being trolled is part of, it's like part of life now. Now, trolling can occur in 3 levels. One, it can come from someone who is a universal troller. For example, if you use certain terms, certain medications, we use the certain words. For example, if you even say the word ivermectin on social media, you'll be trolled immediately by people who want to push that drug on you. And if you say something against it, they'll come and attack you personally. but you need to find a way to defend those. So that's one. So those you can ignore. And then there are some trolls who don't understand what you're trying to say. And then they may give you a slightly offensive outlook, but they may not be as bad as you think it is. But if you give a firm, assertive note, I pretty much give them the same force that they gave me. And that's the technique I use. I estimate the force they use and I give them exactly the same amount of force. Never foul, but forceful. And they will understand and they will settle down. And then the third category is people will ask you questions. You're not obligated to answer any question for anyone because it's your time. But if you want to, you can answer them. And I think if you answer questions well for someone, it may help other people who are silently reading the timeline. So I do that a lot. And in the beginning, I remember in my Twitter, there were very few people who would even look at my handle. And I was wondering what is wrong with the world, because I was giving the best possible information out of hours and hours of hard work and not even one person looking at it. But I realized that if you're there long enough, if you engage and if you keep posting content which is good, yes, people look at it. But I don't measure success by number of followers or anything. In fact, I don't have a large number of followers either, because I don't lobby for that. I just put out science. I just put science out there for people who want to look at science. So that is what I do. That's what I stand for. So I can't speak for everybody, but I think my messages would be, if you're consistent and pure in intent, I think you'll be able to get through whatever system you're working in. If you work in the government, if you work in administration, I think it'll happen. But then there are some people who want to profit during the pandemic, and I don't support that at all. And there are plenty of fake treatments that people are dishing out, fake claims, and so on. All that's wrong. You heard about the lab in Manjeri that is giving out fake certificates without even testing an RT-PCR. And all these things are happening, and I think we should even raise a voice against these tendencies. Nobody should be allowed to profiteer unfairly from the pandemic. And so these are some things that I have to say. I've skipped long COVID because it's a long topic. For the most part, I've skipped in terms of pediatric COVID, again, because I'm not a pediatrician, and talking about children and COVID takes a long time. It'll take at least 15 more minutes, so I'll skip that. So
Dr. I think I have a question for you. We all know that in the first phase of infection, Kerala has successfully controlled the COVID cases in a very good manner, especially home quarantine and good monitoring from the local bodies, as is suggested even from the Anganwadis or whatever. There's a good kind of a controlling. But then,
do you mind turning your video off? I can't hear you. Sure. Just turn your video off so I can get your audio well. I want to hear you well.
Yeah.
Okay.
Okay, can you hear me now?
Slightly better. Yeah, tell me. Yeah.
Okay. I was just referring that last year Kerala has controlled COVID in a very good way, especially all over the places especially from international and national community, that on home quarantine, the success of home quarantine and controlling the cases thereby. But recently, there was a report the health minister herself told that now the infection is spreading inside the house.
Okay, I get it. I get it. All right.
Yeah. So which is accounting why one of the big reasons why we have a high case load now. Probably this is also related to the high testing and more surveillance, not surveillance per se, but more testing also to do with the high case.
It's a good question. So I'll summarize the question in just one sentence. Why is the spread more in homes now compared to last year? Right. So the answer to that is that compared to last year, our contact tracing system, it is impossible to maintain that level of contact tracing. See, contact tracing basically means I know all of you know this, but I'll take you through the steps. If I am positive, I need to alert the health services. The health service have to come and find out my contacts, and they have to go and test those contacts based on their risk status and so on, and their contacts and so on. So remember the Nipah case in Cochin that occurred in, I think, a couple of years ago, and close to 500 contacts were tracked down. That takes a lot of effort, right? It's not easy to track down that many number of contacts. And that was done last year with a lot of enthusiasm. Why? Two reasons. One, it was rare. The disease was rare. The danger was unknown. We never knew what the ending was of that particular wave was. And then people were, it was like Nipah reloaded for them. So they were all excited. They wanted to contribute. Everybody wanted to contribute. So everybody was enthusiastic about this. And there were only few cases to track down. See, it's very easy to create a contact list of 50 if you have just 10 cases in your town. But now if you have 20,000 cases, you can't really do that. It's physically impossible. Where are you going to get the manpower? The manpower is the same. Same healthcare workers, same ASHA workers, same JPHNs who do the same work. And they're all into vaccination now. So where are the people? We can't create people out of thin air. So contact tracing could not be done. And that being said, I'll also say this, when there are 20,000 cases a day, the role of contact tracing may not be much compared to there's only one house or two houses in a town that are infected. Yes, you must do aggressive contact tracing, try to contain the disease. But when the disease is everywhere, contact tracing does not have that much of a relevance with limited healthcare resource. The second point is within homes, the virus is spreading because half the transmission is asymptomatic. Delta spreads faster and infection starts spreading 2 days before symptoms. Even in the half who develop symptoms, they've already let the virus out to people in the house. So that is really the answer to the question, why is the virus spreading in the homes? So we are at 3 o'clock now, Dr. Shirley.
So thank you, sir, for this session, and we'd like to have you back again. If possible, do continue joining in for Melvin sir's talk also. So
I can't hear you. Are you muted?
Yes, sorry, sir. Yeah, so let's move on to the next talk of the day by Dr. Melvin. So Melvin sir basically did his MBBS, MD and DM from JIPMER. Sir's area of research includes cardiovascular disease, COPD, clinical trials, evidence-based medicine, pharmacogenetics, bioethics. Sir has with him a lot of awards, especially in relation to grants from agencies like ICMR, MCI, and sir has delivered multiple guest lectures in more than 40 national workshops. And sir is the investigator in Covaxin phase 1, 2, and 3 clinical trial. He is investigator in the TIPS 3 Odyssey outcomes trial. Sir has more than 65 research publications, 628 citations, and book chapters 8 in number. So I request sir to give a talk on the clinical trials of COVID-19, especially the methodological aspects. So over to you, sir.
Yeah, thank you, Dr. Shirley. At the outset, I wish to thank the organizers of Bioradiance for giving me this opportunity to come and speak to you about this clinical trials with COVID-19 vaccines. I hope my screen is visible to you.
Yes, sir. Please carry on.
So we all know that presently this is what is happening across the country. Vaccination status as of yesterday is 77 crore doses have been given, out of which only 19 crore people have received 2 doses of the COVID-19 vaccines. Having said that, it is really something, a remarkable achievement that we can say which has happened in the last year. That is because in a span of 1 year, we have been able to identify the cause of infection and also bring out a vaccine in a record time of 11 months when Pfizer and Moderna came out in the early December. And we should also appreciate the efforts led down by the scientists in India to bring out the vaccine, the Covaxin, in a record time of even 1 year. And thanks to this, we have been able to make some headway in giving vaccines to even our own population, even though we're still far behind in giving it out to the majority of our own population. But it is no mean achievement to bring out such a vaccine at such a short time. How have we been able to do this vaccine development at such an accelerated phase? If you look at conventional vaccine development, it easily takes roughly around 8 to 10 years. Now, what we have seen happening here is a kind of seamless clinical trials where phase 1, phase 2, or phase 2, phase 3 goes on parallelly. And as a result of that, we have been able to do this whole process of something that happens for as long as 8 to 10 years. We have been able to bring it out within 1 to 1.5 years. So that seamless kind of development, which is usually not acceptable. Usually the DCGI, the regulator of the country, they expect all preclinical data to be submitted prior to starting phase 1 clinical trials. After completing phase 1, you start doing phase 2 clinical trials. After completing phase 2, you start doing phase 3 trials. But now, because there was a desperate need to develop a vaccine, the regulators were able to accept parallel development. That is, even as preclinical studies were being undertaken, phase 1 and phase 2 started happening concurrently. And before even phase 2 could complete, the phase 3 trials had started. And this kind of seamless development is what really helped the innovators of various vaccines to bring out their vaccines in the market. So what I'm going to talk about for the next 30 minutes or so is about how vaccine clinical trials are done for COVID-19, how they have been done. And I'll talk about the guidelines issued by the CDSCO for doing these kind of trials. And after that, I'll probably take a few questions if we have any. So if you look at phase 1 clinical trials, now when you say phase 1 trials for COVID-19 vaccines, they are essentially done to identify the safety of the vaccine, where we compare different doses to find which is the dose which is safest and at the same time does not produce much of reactogenicity, no reactions, no allergic reactions. And at the same time, we also try, since we are doing this trial, we also want to capture immunogenicity data. That is, how much of antibodies are developed following vaccination. And the present guidelines state that for every vaccine that is discovered in India, you need to start from phase 1 clinical trials, and subsequently, either you can go sequentially or through seamless development, you could proceed to the next phase of clinical trials. Phase 1 trials are almost exclusively done in healthy volunteers. There's a very stringent inclusion-exclusion criteria to really see that these are healthy. I remember last year when we were doing the Covaxin trial, it was very challenging because one of the criteria said that the person should not have been even exposed to COVID-19. And so even if someone is antibody positive, he becomes excluded from the clinical trial. Apart from that, even investigations like LFT, RFT, or even the blood investigations, almost everything we should see something where a normal picture, only then you will be able to recruit these patients in the study. So phase 1 trials, almost you would not see people with people who are having high risk, who have high risk to develop the infection. Such kind of people are usually you avoid taking them in phase 1 clinical trials. Coming to phase 2 trials, in phase 2, the goal is primarily to assess the immunogenicity component of the vaccine. And of course, we also see the safety. It is recorded because this is going to be done in a slightly larger population. Usually phase 1 and phase 2 in COVID-19 vaccines, by and large, we see trials, the numbers are somewhere between 300 to 700. So we would look at immunogenicity as well as safety here, with immunogenicity being given more importance than safety. And this is also done in healthy volunteers, and the intended population is tested. There might be expansion of age groups For example, in Covaxin trials, we were able to see that the trial was done in a slightly younger age population, almost 12 to 65 years, whereas in phase 1, it was restricted to 18 to 55 years of age. And in phase 3 trials, when you talk about phase 3 trials, you are essentially talking about efficacy, the most important component of the vaccine, as well as safety. The reason why safety is also important here is with phase 1 and phase 2, you're doing the trial in a very small population of, say, a few hundreds. So you would not be able to capture rare side effects, and that can only be captured in a large phase 3 clinical trial. So safety is essentially important in phase 3 also, and efficacy in terms of clinical outcomes are being evaluated for the first time in phase 3 clinical trials. And sometimes they could also consider taking a subpopulation and doing antibody testing in them to see immunogenicity data even from phase 3 clinical trials. And in phase 3 trials, you also notice that most of the sponsors, they tend to choose population which are at risk as one of the subsets, if not the entire population, at least one part of the study, or maybe they might do it in 2 groups. One is an open group having all people, and one is a group which is high risk group. They would say at least 30% of the total sample size will be at risk population. So this is something that is done in phase 3 clinical trials. Emergency use authorization, almost most of these vaccines, emergency use authorization was given, and it was just on the basis of the interim analysis of the phase 3 clinical trials. So now let's look at one particular vaccine that was developed in Australia, for which a trial is ongoing. So if you look at the phase 1, this is a phase 1 trial. So sometimes you would even see a situation where it's not even done in a randomized clinical trial. It's a non-randomized clinical trial, because the goal here is more to see the safety, reactogenicity, and immunogenicity, because this is the initial data that is being tested, where we are trying to obtain this data and compare between intramuscular and intranasal routes of administration for a new vaccine. So if you see the outcomes here, in this phase 1 trial, the outcome was primarily the safety data. Okay, we were looking at solicited local adverse events. Now, what is a solicited adverse events? We look for events which are expected to occur. Okay, so pain, redness, swelling at the site of administration, nasal discomfort, stuffy nose, loss of smell, sore or scratchy throat for if a person is having intranasal administration. All these side effects are something that are specifically looked for. And what is the severity? What is the incidence of these adverse events? And a humoral response to the SARS-CoV-2 antigen as measured by neutralizing antibody assay. And the secondary measures, we looked at again humoral response to the SARS-CoV-2 spike protein as measured by IgG and IgA, and mucosal IgA antibody response, that is for especially the intranasal vaccine, and measurement of cytokine producing T cells specific to SARS-CoV-2 spike protein, and measurement of the adenovirus neutralizing antibodies. So this was something that was seen in a particular phase 1 clinical trial. So you can see here, there's a mixture of safety data being collected and immunogenicity data that is also being collected. So clinical trials are, if you look at particularly the phase 3 clinical trials, either they are expected to demonstrate a superiority design or a non-inferiority design. So if you are having a placebo-controlled trial, then you're obviously talking about superiority trial, where you expect the efficacy to be better in among the people who take vaccine. You are expected to see lesser number of infections or lesser number of RT-PCR confirmed infections or clinical infections. Now, if you are talking about a situation where you already have another vaccine, okay, and so you're comparing it to the other vaccine, then even you could consider doing a non-inferiority clinical trial where you have a non-inferiority margin that is spelled out right at the beginning, so that you are able to show that the new vaccine is not inferior to the existing vaccine in the market. So it's enough if it is able to have an efficacy that is more or less within that margin that is claimed. In most of these clinical trials, in the early phases, especially phase 1 and phase 2, they generally try to avoid taking people who are at a high risk. One of the main reasons is because there is always this theoretical risk of vaccine-associated enhanced respiratory disease. This has been something that has been seen with earlier vaccines. So even though we have not seen that with COVID, to the best of what I've seen in the literature, it has not been reported, but this risk is always there, and that is why they do not want to take these vaccine for early phase vaccine trials. They do not usually take the patients who are at high risk of infection. And even older adults can participate in early phase clinical trials, but at the same time, it's essential that such people, if they're older adults, particularly, they should not have any medical comorbidities. Children, if you are going to take if you are going to take people, that is older adult participants for certain vaccine platforms where you do not have any experience with that particular vaccine platform, then it is better to start with younger adults first, get some basic data where you're getting at least safety data for a preliminary period of at least 7 to 14 days. Get preliminary data and then start considering older adults. This is, I'm talking about a situation when you're using a new platform that has never been utilized. Now, if you look at Covaxin, it is not a new platform. It is something that is well known, an age-old technology, inactivated vaccine. So obviously, this may not apply to that. But if you're looking at something like a DNA vaccine, something which Cadila has brought out, then you would have to consider this particular clause. And in healthcare, workers are also traditionally not taken up in early phase clinical trials. Okay, the healthcare workers, if you see Covaxin or even if you see the other clinical trials, they are taken much in the later phase of clinical trials. But it is essentially important to gather data about these populations also subsequently. Can people who have past history of COVID-19, can they get themselves enrolled in these vaccine trials? Now, the answer to that is because people who are going to be ultimately vaccinated, it is not practically possible to sit and check to see whether they had COVID-19 or not and all that. And because it is such a common, COVID-19 is so common, it is almost universally recommended that not, at least in phase 3, to not limit ourselves to excluding these people from the study. That is, it is encouraged that we include people who also had past history of COVID-19 so that we are able to see how these people respond to the vaccine in comparison to those who didn't have COVID-19 history. Okay, so subjects with lab evidence of prior COVID-19 infection should not be excluded. They should be included in the study, particularly in the later phases of the clinical trial. And if a person has acute infectious illness of any kind or even COVID-19, obviously they'll have to be excluded from such clinical trials. And it's also important to look for diversity. So we have to take as much as possible a diverse population. The population not be too homogeneous, because then the results may not be applicable to across all populations. So pregnant women traditionally are always excluded, lactating women are excluded, though there is a guideline stating that if you have good, adequate data from the reproductive and development toxicity from animal studies, then you could consider including them. But even though this is there, practically speaking, there is in clinical trial scenario, at least usually they are not taken. Usually they are excluded from clinical trials. And most of the recommendations right now we see are more based on experience in the post, that is in phase 4, and when it goes to the market, and gynecologists are starting to use them in practice, and then they have seen it is relatively safe and it can be taken. So many of the ACOG and many other societies have now recommended giving them even in pregnant women. So what about blinding in early phase clinical trials? Now, it is not mandatory, obviously, in early phase trials, but if you are doing blinding in early phase trials, it is certainly going to help if you're looking at safety data, okay, because there is going to be a lot of bias if there is no blinding. And even in terms of holding a patient for the throat, because most of these clinical trials will have some degree of follow-up. If there is no blinding and if they are all aware that they are receiving something like placebo, then it is not going to be giving much, they're not going to be very compliant. Whereas in late stages of clinical trials, that is phase 3 trials, it is encouraged that randomized double-blind placebo-controlled trials are done so that you are able to get robust data, and particularly because in late phase trials, you are dealing with clinical response rather than immunogenicity. So since clinical response, that is clinical, they are going to report the infection back to us on the basis of what they say as that they have got the infection. You're going to decide whether they have attained the primary outcome or not. So if it is double blind, obviously it will be having less amount of bias. So classically, we see a vaccine is compared with placebo. That is the most efficient design for demonstrating the efficacy of the vaccine. But there is a guideline. It also tells that if you have a situation where you want to compare multiple vaccines, then you could go ahead and compare multiple vaccines at a time with placebo. So this is there in the guideline, but practically speaking, it has not been something that has been followed. Even though it is very efficient, practically almost most sponsors will look at a single dose compared with placebo. So now the next question that you might think is, can we use placebos now, considering the fact that COVID-19 vaccines are already available in the market? So the guidelines clearly say that once vaccines become available, then you should always look at giving vaccines as controls and not placebos. In fact, it was very surprising that WHO has mentioned that in their ad hoc expert group that you can, in developing countries, because of COVID-19 vaccines are not available, placebo-controlled trials can continue. There was a lot of questions raised regarding this from several corners. And if you see even some news that happened, I think a month back, 2 months back, Covovax phase 3 clinical trial, which Dr. Gagandeep also mentioned. This study was originally supposed to be a placebo-controlled trial, and then subsequently it was changed. The placebo component was removed, and so all people were receiving the vaccine. So as of now, if you write any clinical trial and you ask for a vaccine placebo-controlled trial, you're unlikely to get a DCGI approval for such a kind of clinical trial. How long should patients be followed up after a COVID-19 vaccine trial? So conventionally, what we see is patients are usually followed for a period of 1 to 2 years, because we do not know how much time it might take for the immunity to sustain. So traditionally, one year at least minimum, and 2 year would be even better to assess duration of protection and potential against vaccine-associated enhanced respiratory disease once the immunity. Now, we might have another common scenario, which has already happened, even in the case of Covaxin, is you're doing a vaccine clinical trial, and midway, that particular vaccine gets approved on the basis of preliminary data, interim analysis, and it gets approved in the market. So then there are a number of people who are, or you can say almost half the people in the trial have already received placebo. Now, what happens to them? The answer to that question is, now the recommendations say that clearly you should consider stopping them and having a consult CDSC office and start unblinding these people who have received placebo, and then call them to the study site, give them the vaccine. So this is exactly what was done even in the Covaxin clinical trial. Patients who are receiving placebo were asked to stop after a considerable degree of the trial was over, and they were given vaccine so that they are not denied the benefit of getting the vaccine. Are there any geographical considerations in deciding for a sponsor that which are the clinical sites, which are the areas where clinical trials for the vaccines need to be happening? Usually, all as you know, all these vaccine trials are done across several centers. If it is a trial happening in India, there'll be at least 10, 12, or 15, 20 centers. So which area to do the clinical trial? So the answer to that question is that they usually choose a center which is having a high degree of infection, where the incidence of the infection is high. So that there is a quicker chance of developing the outcome so that they are able to complete the trial. So that is one of the major criteria, besides the other usual criteria that the site should be having investigators who are experienced, trained, who have previous experience working with vaccine trials. Those are the usual things. But apart from that, for COVID-19, this is one important criteria. So what is the threshold that has been suggested by WHO is at least 50% efficacy should be there. And as ma'am said in the beginning of the symposium, that almost all the vaccines which are currently there in the market have crossed this particular threshold. And the common endpoints used in the vaccine phase 3 trials are a laboratory confirmed COVID-19 infection. And besides that, some are also looking at collecting data of asymptomatic infection. Okay, so how much can the vaccine prevent asymptomatic infection? How much can the lab prevent infection, that is clinical infection, where the patients have the symptoms? Okay, and besides that, you might also think that ultimately, when it comes to COVID, they are most bothered about the severe infection. So why aren't trials designed to look at that as the efficacy outcome? So the answer to the question is, obviously it is important. It is in fact the most ideal endpoint. But the problem is, if you use that is prevention against severe disease and death as your endpoint, what will happen is, since that is not such a common outcome that occurs, what is going to happen is your sample size is going to be unfeasibly large. So you're going to need almost lakhs of individuals in your trial to complete the trial. So as you can see here in this paper, the sample size required, okay, it says if 0.2 infections per 1,000 people develop over 6 months, if you use infection as the endpoint, just infection, that is without even symptoms, then you would need around 1880 patients. If you were to use symptomatic infection, you would require around 3,154 patients. Whereas if you were to look at hospital admissions, from 3,000, it has run up to 1,83,000. And if you were to take death, almost 6,19,130, 6 lakh individuals you would need. So this is the point that we're trying to say, that if you're taking hospital admissions, prevention of hospital admissions and prevention of death as your primary outcome, then you would need a very large sample size. Unless you're doing the trial maybe exclusively in elderly individuals where the risk of this outcome is very common, then it would not be feasible to do such trials. So then how do you get this data? How can the effectiveness of vaccines be assessed against severe disease? So the answer to that is do epidemiological studies, post-marketing studies once the vaccine is deployed in the market. And we already got some kind of data for that has been alluded to by the previous speakers. So that's how we have known now it is good enough. And another question is, okay, a lot of clinical trials have been published. Are they really reflecting the real world scenario? The answer to that question is yes. There have been instances in the past also where vaccines have been found to be very good, okay, in trials, in the low income, middle income settings, and but at the same time, it was not as high as the efficacy observed in high income countries. So this kind of things have always been there in clinical trials. What we see in trials and what we see in the real world does not always match. And that's why we always look at real world studies done after the drug or vaccine is launched in the market. And with vaccines, there is one more important thing to consider, and that is the herd protection. Okay, so clinical trials will never assess this herd immunity, because in clinical trial you're only looking at the efficacy achieved for that particular individual. Whereas when you're deploying the vaccine across the population, there is going to be a bulk of the people who have not received the vaccine, but who are still inherently protected because the people around them have all received the vaccine. So, and naturally, the risk of transmission reduces, and the risk of them developing the infection also reduces. So these things cannot be assessed by RCTs. Now, I was talking about high-risk population. It is important that when you're doing a clinical trial with COVID-19 vaccines, you take a particular percentage of the patients to be from this population, because only if you take them, you're going to see those people, a considerable portion of your trial population, developing the outcome. Particularly, imagine a scenario in India right now. Imagine a trial is happening right now in India for COVID-19. It will not be easy for the sponsor to develop outcomes. God forbid if the third wave happens. In such a scenario, you will not be able to get enough outcomes. So what will happen? You will have to do the trial for a longer time and wait for more number of events to develop. So the solution to that is take people who are high-risk population so that you are more likely to develop the outcomes. And sometimes you would also have a situation where you can develop vaccines which may not affect clinical course, but it reduces the transmission of the infection. Even that at the population level is a very suitable option. So, some of the practical difficulties and challenges in recruitment of individuals is when you're, suppose even if you're taking elderly. Now, elderly, obviously, having them because they are at high risk, it is good to have them in the trial because more likely to develop outcomes. But at the same time, it is the same elderly who are also people who tend to shield themselves by avoiding social gatherings or use of public transport compared to the younger individuals. So then developing outcomes in them sometimes may be limited. Similarly, healthcare workers, if you see, on one side, they are at a high risk of infection because they are getting a much higher dose of the virus. And so they are at high risk. But if you look at protective strategies, they are the people who are more likely to use protective strategies. So having them in the trial also has this thing of not developing the outcomes because they are using multi-pronged approach to preventing infection compared to the general population. So these are some challenges involved in recruitment. And I think I'll skip this also. So another problem with incidence of, within SARS-CoV-2 vaccine trials is that the infection, that is the COVID infection incidence, is highly variable. It is not something that is static. So when you're doing the sample size calculation, you calculate based on the incidence, okay? You make decisions based on the background incidence of the infection. And then halfway down the study, you see that the infection prevalence incidence has totally changed. So then it becomes very, very difficult for you to continue the study because then your sample size calculations, everything will go for a toss. So this is another common challenge that is there, the variable incidence rate. So as the incidence of this SARS-CoV-2 decreases, the time required to meet the efficacy endpoint keeps increasing. That is a common challenge in clinical trials with COVID vaccines. So this, I think I told you about site selection. So one way of picking up these mild infection is to have a process by which you serially collect samples of patients. You ask them to come maybe every week or every month, take a sample by default, and ensure that you do not miss out asymptomatic infections. Okay, in that way, you are taking asymptomatic infections as well as people who obviously report, because people who have this tendency of not reporting minor infections. If they get a fever or a cough, cold, obviously they would think it may not be COVID, so why should I report? So that is the general attitude people have. So ensuring that they come for regular sample, and they provide regular samples is one way to pick up these kind of infections, which could otherwise go undetected in phase 3 clinical trials. Now, it is possible that when you're doing a clinical trial, phase 3 clinical trial with COVID vaccines also, SAE happens. What is an SAE? A serious adverse event, something which causes death, hospitalization, or it causes disability. Okay, so in these situations, the way you handle an SAE is almost similar to how you handle SAEs for all other clinical trials. The first thing you have to do is the medical management. You have to take care of the patient, the participant, trial participant, ensure that when he's there in the hospital, his medical treatment is taken care of. Now, the next question you have to answer is, will he continue in the study? So usually what they do is the person would not be continued. Generally, they are not continued in the study. They would consider sometimes doing a unblinding of the participant to see whether they receive the vaccine or the placebo. This will help them to make a judgment about the causality analysis, whether it is a vaccine-induced problem or it is something that has happened naturally because of other factors. And if it is proven that it is related to vaccines, then obviously the ethics committee would write this recommendation to the DCGI stating that this is more a vaccine-induced problem, and so the sponsor has to compensate the patient who has sustained a serious adverse event. The DCGI will review their own ethics committee. Their DCGI will recommend to their own expert committee. That expert committee will take the final call and submit it to the regulators. The regulators will recommend to the sponsor to pay compensation to the patient who sustains a serious adverse event that has been proven to be because of the vaccine. So this is essentially what happens in a serious adverse event scenario. Some of the other challenges that are faced by sponsors of vaccine trials, enormous cost to manufacture, especially if you're talking about scaling up post-trial, and we are talking about deploying across countries, and I don't need to say much about risk of politicization of vaccines. This is something that was a raging debate in the beginning of this year. So other trials that are seen by vaccine during vaccinations, most of these trials, you would see that people, the first few visits they come, they are enthusiastic, they come take the first dose, they come take the second shot also, and they might come back for 1 or 2 visits, but then subsequently they get bored because they have their own life and their own things to do, so they might not be that much motivated to come back for visits. So what we need to do is keep reminding them, keep telling them the importance of continuing in the study, because with every dropout, what happens is it becomes more and more harder for us to complete the trial and give good quality data. Another challenge is, of course, getting, as the days progress, as we see more and more trials happening, getting non-vaccinated participants will be more and more challenging. And requirement of licensed vaccination at workplaces may make many people, even though they are willing, because of these issues, they are not able to participate in trials. And unavoidable, because right now we don't have a system wherein they take up a certificate where they take it up that, okay, this person is part of a clinical trial, so we don't need the COVID certificate from him. This clarity is not there. So many people are scared to participate in clinical trials because they feel that they'll be harassed because they don't have a COVID certificate. Unavoidable bias towards clinical endpoints. So that's, I was already telling you that they are unlikely to report every clinical event that happens. Okay, so if they know that their RT-PCR positive, then they will report it. Otherwise, they might think, okay, it's just a fever, why to report? So a lot of subclinical infections or mild infections are going unreported. in the placebo group as well as in the vaccine group. Whether in which it is higher, we do not really know. Participants might be also more likely to report symptoms meeting clinical criteria for COVID-19. So this is what I was telling. So in conclusion, I would say that it is really a remarkable achievement that COVID-19 vaccines have been developed at such a breakneck speed. And currently, there are a lot of new vaccines that are being developed against variants in pediatric population, elderly, and other high-risk population. A lot of trials are underway. And the lessons that we have learned on developing vaccines such a rapid pace, with all the regulatory hurdles, we have been able to overcome all that. It's really a story for the future people who are participating in other non-COVID related research, how to develop medicines, how to develop drugs at such an accelerated pace, so that even other diseases, we are able to overcome these kind of challenges. So with that, I conclude. If there are any questions, I'll be happy to take them.
Thank you, sir, for that very informative lecture. So you have highlighted the different methodological aspects of designing a COVID vaccine trial, and you've also highlighted the challenges which you all are facing. And I have been very fortunate to have you on site for one of the good clinical practice sessions, which we think it's kind of boring, but then sir really drills it in your head what is each part of the good clinical practice. So I'm happy that sir could share his knowledge with us. And here at Pushpagiri, whatever doubts in clinical trial, we go to sir and we ask him regarding it. So sir is our well-wisher as well and consultant also. So sir, there are some questions in the chat box. Shall I just take that? Yeah. First is, what are the specific inclusion and exclusion criteria you consider for COVID-19 vaccine?
So generally speaking, if you're talking about a phase 3 clinical trial, the main thing what you would look at is the age. The age of, obviously, if it is not something that you are dealing in a pediatric vaccine, then first thing is they should be adults, and they should not have received the COVID vaccine in the market, which is already there. I'm talking about a vaccine trial which we are doing now, where already there are vaccines in the market. So they should not have had COVID-19 vaccines in the recent time. And another thing is, because there is a chance of the antibodies from the natural infection to interact with the antibodies that is brought up by the vaccines, many vaccine manufacturers, they do not want people who have been recently infected with COVID-19. So even that is one of the criteria. No recent COVID-19, no COVID-19 vaccines. And the age appropriate criteria, gender is usually both are acceptable. And the exclusions classically would be no active disease. I mean, they can have diabetes, hypertension, or any other disease also, as long as it's not active to the extent require, it is well controlled. It should not require hospitalization. As long as the disease is well controlled, not requiring hospitalization, then they can get themselves vaccinated. And I mean, these are the major criteria that I would say that is common to all clinical trials. And of course, by default, pregnant women, lactating women, or women planning to conceive in the immediate future, all these are by default, they are part of all clinical trials. So this is more or less what we can see in all clinical trials.
So the next question is regarding what are the outcome measures commonly studied in vaccine trials?
Yeah, so if you're looking at phase 1 data, you're mainly looking at solicited adverse events as well as unsolicited adverse events that are reported directly by the participants. Patients are usually called up very intensively during the first 1 week after vaccination to study the reactogenicity also, whether they develop any allergic phenomenon. So that is one thing. And besides that, immunogenicity, that is basically antibody testing, is done at different time points, and they see how much the antibody levels have increased substantially, and whether that is statistically significant increase in the antibody titer across a period of 3 to 6 months. So that is for safe phase 1 and phase 2 trials. If we're looking at phase 3 trials, we are usually what we want to see is, does the vaccine have the ability to reduce the risk of infections? So both asymptomatic infections as well as clinical infections. This is the common primary endpoint that's used. Secondary endpoint can also be reduction of hospitalization, reduction of death, reduction of ICU admissions. But again, the study won't be powered for all these secondary outcomes. The primary outcome will be where the study will be powered.
So one question from my side. Presently, there is a research involving children. The vaccines which are available in the market, they are moving on to target children. So there, what all special considerations are, planned before going in for such trials?
So generally speaking, one general guideline that has been given by even CDSCO is that any vaccine should not be directly, even if it is a new vaccine that is discovered, forget about Covaxin and Covishield, but even if some other vaccine is coming up, they should not directly try to test it in the pediatric population. It should have gone through trials in the adult population. It should have been, you should have generated good data from the adult population and then bring it out in the pediatric population. So that is why now we are seeing studies happening, ongoing studies with the existing drugs, existing vaccines. So primary thing is our experience with the vaccine and with the platform, which is the technology platform that vaccine we are utilizing. That experience is very, very important. That is the most important criteria. Besides that, the other criteria would more or less remain the same in the sense that we would have to look at to see that other active infections which could interfere with vaccine response. And again, monitoring and all that would be almost the same, that we are going to see antibody titers, we are going to see risk of asymptomatic infection as well as symptomatic infection. And secondary endpoints would be reduction of mortality, reduction of ICU admissions, reduction of hospitalizations. Yeah.
Thank you, sir. I think there are no further questions. So once again, thank you so much for accepting our invitation and consenting to speak here. Thank you very much for your valuable time, sir.
You're welcome. Thank you.
Thank you.
Ma'am, you're muted.
Yeah, so we are moving on to the last part of Bioradiance. So I would like to share the winners of the e-poster presentation. First place, we have Dr. Sheena Raj. She is a postgraduate from the Department of Conservative Dentistry in Pushperi College of Dental Sciences. So she's won rupees 3,000 for presenting her e-poster in a conference session in the morning. So congratulations to you, Dr. Sheena. We'll be mailing you the certificate and wiring the prize money. Then second prize winner, we have Dr. Ruby. She's a faculty in the Department of Chemistry, School of Biosciences, Jain deemed to be University. She is receiving an amount of rupees 2,000, and again we'll be wiring you the money at the earliest time possible. So congratulations to all winners, and I move on to thanking all of you all for giving such good questions and keeping the sessions interactive. Thank the speakers from the bottom of my heart, especially Dr. Gagandeep Madam, Dr. Rajiv Sir and Melvin Sir, all of them hearing that students are involved, they all were very ready at the first instance. And I have to thank Dr. Mercy Ma'am, Elshiba Ma'am for contacting the speakers and getting their approval for this Bioradiance. I personally have to thank our director, academics, Father Matthew Marvancherlachan, who's been the constant pillar of support for going on for Bioradiance, and this is our 9th session in 9 years. Even if we had converted it online last year also, and we had such good participation for that also. So I thank all the participants for consenting and coming over and listening to this. And also I thank my colleagues, my friends and students. All of you all have played a major role in supporting Bioradiance, but none of this would have been possible without very efficient support from our MAC first. They have helped us in setting the online platform and conducting the poster competition, everything perfectly. Thank you all so much. I hope you all have received enlightening materials from this webinar, and kindly use this in your day-to-day practice. And if you're researchers, again, you would have got some research question where you want to go in further and come up with some answers. So all the best to all of you all, and thank you once again for participating. So do fill in the feedback form so we can improve and make ourselves much more better the next time. If any suggestions, we are welcome to listen to it. So thank you all. So bye-bye. Good evening and have a great day ahead. Thank you.